start about the female reproductive system, we have few content to cover in the female reproductive system. Just like male reproductive system, we only focus on testicles or microanatomy of testis. In case of the female reproductive system, we have number of organs to cover, like ovary we have to cover, which we are going to do today. Then we have this uterus and the fallopian tube, which is as well equally important like ovary as a microanatomy slide is concerned. So, whenever you are going to study the ovary, likewise this testicles, we are going to study this particular different components and parts, which helps us to identify or separate out ovary from the rest of the structures. Now, as we know, this particular ovary is an organ of ovulation, but having that particular part as well, in addition to that ovulation functions, the ovary also performs as an endocrine organ. So, it is having a dual role, and that particular entities we are going to address whenever you are going to identify that in microanatomy of the ovary. So, this particular tissues or the ovary, whenever you are seeing that, is covered by a thin layer of peritoneum. That particular peritoneum got little bit modified and covering it. That covering we call them as epithelium, and it is known as germinal epithelium. But having said that germinal epithelium, it does not have any role in germination or productions of the germs or gametes. So, this particular outer layer of this ovary covered by a single layer of cuboidal cell epithelium, we call them as germinal epithelium. Now, if you go deep to that, we will find a connective tissue layer, we call them as tunica albuginea. Now, within this particular tunica albuginea, which is spreading out from this particular outer side to inside, we divide this particular ovary parenchyma into two parts. Into outer side we call them as cortex, and inner side we call them as medulla. The medulla side is predominantly occupied by the presence of the blood vessels, lymphatics, and as well as collagen tissues, whereas the cortex is occupied by the germ cells. Now, in case of these germ cells which is present in ovary, we call them as this particular. In case of the female, we call in case of the ovary, the germ cells we call them as in the various stage of follicles, we call them as ovarian follicles, which contains a cell within with this nucleus, we call them as oocyte, which is basically germ cells. Now, this particular different various stage of this ovarian follicle, that we have to understand what do you mean by the different stage of ovarian follicles. Now, if you see this particular schematic picture, this is a flat spread out picture of the ovary. Now, if you see that this part, we call them as surrounding epithelium, we call them as germinal epithelium. It is a single layer, simple layer of flat cuboidal cells which is covering to it. Now, deep to that we have this particular tissue, we call them as tunica albuginea, which is another layer which is lying there. Now, this particular whole thing from this side to this side, we call them as parenchyma. This parenchyma is divided into two parts. This is the outer side, we call them as outer cortex. This is the central side, we call them as medulla. So, medulla is occupied principally by the blood vessels and collective tissue structures, whereas the outer cortex is occupied by the different stages of ovarian follicles. So, we have these different stages of ovarian follicle that we have to understand what are different stage of ovarian follicles. For that reason, we have to understand how this particular ovum or oogenesis process is going on. So, we get this particular germ cells in the ovary coming from these areas of the developing embryo, we call them as yolk sac. So, from where these particular cells which migrates ascending through this mesentery, it reaches to these gonads. Once it reaches to the gonads, we call them as the germ cells. Now, depending on if you have X chromosome or Y chromosome in our system, that particular germ cell transforms into that particular gonad transforms into the testicles or ovary. Now, if we have two X chromosome, that particular gonad transforms into the ovary. If you have one Y chromosome, that particular region of the Y chromosome, we call them as SRY or sex determined region of the Y. That particular Y chromosome determines or transforms the gonad into the testicles. That is how these things are converted into either testicles or ovary in the subjects. Now, once this particular germ cell reaches to this gonad or primordial gonad, based on this X chromosome, this particular thing transforms into ovary, and this particular cells, germ cells, goes in a mitotic divisions, which is leads to this formation from the primordial germ cells into oogonea, and then it goes to a next stage, we call them as primary oocyte. Now at this particular stage, this primary oocyte, the cell division stops. We call them as the prophase stage of diplotin substage. We have the arrest of this primary oocyte development. So it stops at that particular age. Before we are born, this particular things is happening, and it remains at this stage till the puberty, till the hormone comes in. Now in the diplotin stage, it is arrested in prophase 1. We have this primary oocyte. Now this primary oocyte is covered by a layer of cells, we call them as follicular cells. So this what we are going to talk about, different stage of maturation. Now you see that we have these different shapes and different size of these developing follicles are there. So this particular follicle, you see that this is the tiniest one compared to this is the largest one. So we have the different stage and intermediate stage which is present like this one, and a terminal phasing out phase which is present in here. We do not see any kind of like a nucleus or oogonea into this stage. So we have the different stages of this maturing ovarian follicles present in the cortex, and this particular thing have got a different stage of life cycle this is going through. Now this one we are taking up the smallest one, we call them as, this one is primordial follicles. Now what is this primordial follicle is composed of? It has two elements. One is this oogonea which is present, the nucleus which is present into it, which is arrested at this particular mitotic 1 diplotin stage, and it is covered by a flat single layer of cells, we call them as follicular cells. So this consists of these two components, we call them as primordial follicle. Now this primordial follicle matures into next stage, we call them as primary follicle. What happens to this? This particular flat cells becomes cube cells, becomes fatter, get some kind of nutrition to it, becomes fatter, then we call them as primary follicles, and the size of the oogonea also enlarges, but it remains on the same developmental stage of maturity 1 on that particular cells. Only the size increases, but the cell division was arrested and remains arrested. Now this particular region we call them as primary follicle. We have more than one thick layer of these flat cells which become thick layer cells or cube cells surrounding this, we call them as primary follicle. Now after this particular primary follicle, it becomes secondary follicle, which is this one. You see that this particular cells become stratified, this one cell becomes multi cells, and within this particular multi cell it distinguishes between this particular outer layer and inner layer. And into here we get a layer, we call them as this layer which is surrounding this oocyte, and into there we find a cavity to it. We call them as antral cavity. So you see that this is a like early stage oocyte surrounded by this particular flat cells. This particular flat cells become this square cuboidal cells. We call them as primary follicle. Now after this primary follicle, it gets a kind of glycoprotein coat inside to it. We call them as zona pellucida, which covers between this particular oocyte and this particular follicular cells, which is present in it. Gradually, zona pellucida surrounds to it. You will see that the next stage, the primary follicle moves to the secondary follicle. The size enlarges, the cellularity increases, but this particular oogonea remains at the same developing phase. Only this particular oogonea gets in a bigger size, and surrounding by this particular flat follicular cells, we call them as flat follicular cells as granulosa cells. Now this particular granulosa cells and the theca cells, these two transformation happens within this particular follicular cells. The cells which are present outside, we call them as theca cells, which are present surrounding close to this oocyte, we call them as granulosa cells. The theca cells also differentiates into two subgroups. We call them as theca interna and theca externa. Now around this particular developing oocyte, we have a cell layer existing to it, which remains as a radiating fashion of the follicular cells. We call them as corona radiata, and this particular layer is covered by this particular glycoprotein coat. We call them as zona pellucida, which is very much present. And after this, we get a cavity which is develops within this particular follicular cell layer. We call them as antral cavity, which initially was small and sporadic, later on becomes big and large, and then it covers whole of this particular region and makes a big cavity. We call them as antral cavity, and this antral cavity gets filled up with the fluid substance. We call them as antral fluids, which is present in it, and pushing this particular oogonea into a side like this and surrounding by a glycoprotein coat. We call them as cumulus oophorus, and a radiating cell which is covering here, we call them as corona radiata, push to it and becomes more and more mature. We call them as mature follicle or Graafian follicle. Now, this particular Graafian follicle with presence of some hormone bursts open and ovulation happens. So, after the ovulation, this particular follicle becomes a collapsed follicle. We call them as degenerating follicle. Based on how these particular degenerating follicles are there, in a stage we call them as corpus luteum and corpus albicans. First it becomes corpus luteum, so it occupies some pigments inside. That particular pigment thing looks like yellow, we call them as corpus luteum. And then afterwards, after this particular corpus luteum, which is very active hormone producing organ, becomes a scar tissue as a white tissue, we call them as corpus albicans. So, this particular phases of different maturation happens to occur in this particular developing follicles, and we call these particular phases or different phases of follicle can be seen within this particular cross sections of the ovary. So, some of the follicles we may see in a primary stage or primordial stage, some of them are present in this particular like primary follicle stage or secondary and Graafian and ruptured. So, all these particular phases can be seen as a kind of like a cross sectional picture whenever you are examining this particular histology of this ovary. So, this is what you have to keep in mind in regard to this particular follicular phase. We call this as a follicular phase from the primordial follicle to primary follicle to secondary, tertiary, which is going to mature eventually to the Graafian follicle, which burst open into the ovum, and finally it transformed into the corpus luteum or hormone producing organ, then corpus albicans as a scar tissue and regresses out completely. Now, during these particular phases, we are going to identify different phases of the ovum or different phases of this particular ovarian follicle. We have two of the specimen which is good in a sense visibling. We have this particular Graafian follicle, one of them, and couple of them we can see this particular primary and secondary follicle as well. So, whenever we are given this particular histological pictures to identify them, we are going to identify this particular ovary based on these particular regions, like it is having a outer layer covering, we call them a germinal epithelium, which is a flat kind of like a cuboidal epithelium. Then we have this parenchyma, outer cortex and inner medulla, which is mostly occupied by the connective tissue and blood vessels, whereas the cortex have got different stages of maturing ovarian follicles, which is present in different phases, primary, secondary, primordial and Graafian, and as well as the scar tissue, etc., there. So, this is how it should be schematically looking like, but whenever you are focusing to this particular film, you may not see all these things, but couple of them will be focusing on. So, we are, if you get a kind of ripe follicle, we call them as Graafian follicle like this, we will focus on the Graafian follicles like this, which is going to get mature follicle and eventually going to be the ruptured one. And of course, we are going to identify this particular epithelium and cortex and medulla whenever this particular tissue slides are there. These particular phases and stages are important for us. This particular stage or tertiary follicle or Graafian follicle. Sometimes we get a clinical questions based on this particular Graafian follicle. They ask you to draw a schematic picture of this particular matured ovarian follicle with different pointers to it and identify the different phases and how these particular stages are there, which is coming to it. Now, we have to discuss about two entities whenever you are going to discuss about. We have to talk about the oocytes and different stages, we call them as follicular stage, which matures into different phases into which is present in these particular regions, like different entities, like we have to talk about this particular region, we call them as the glycoprotein coat, which is present inside. We have to talk about this particular region, we call them as like corona radiata. We have to talk about theca cells, we have to talk about theca interna externa cells, at follicular antrum or antral follicle filled up with the substance, and oocyte, at which stage this particular oocyte is arrested in this particular like a developing phase. After this particular secondary oocyte, primary oocyte, the same thing happens with the primary oocyte, going to become this particular Graafian follicle and expelled out. It completes this particular meiosis 1 and remains there. If we get this secondary oocyte which is expelled out with the first polar body, which is expelled out from this particular matured oocyte, gets these things fertilized, then the further stage of maturation happens to it. If it is not, it is absorbed or destroyed at the secondary oocyte stage or second polar body, first polar body stage. Only after fertilization, this particular second phase of this meiosis 2 happens. After that only it progresses to this meiosis 2 and complete cell separations or nuclear separation occurs. If not, at the stage of meiosis 1, how this particular like secondary oocyte is liberated out, that is destroyed likewise. So, this particular region is point of interest for the short note is this Graafian follicle. You have to prepare this particular Graafian follicle, and this expelled out ovum with different parts we have to know about, like ovum which is present, we call them as female pronucleoside with the first polar body into it, covering here glycoprotein coat which you seeing here in the blue region, we call them as zona pellucida. Surrounding this particular cells to this particular region which remains in a radiating manner, we call them as corona radiata. These are the modified or transformed follicular cells who takes this particular cuboidal to this amorphous shape which is present to it, and this particular whole things travels together. If only fertilization happens, then further cell division continues to go into the like a second polar body formations and completes this particular complete thing. If not, this whole thing is destroyed or phagocytes. So, different stages of this particular ovarian follicle, primary follicle which is having oocyte which is a particular shape. This particular shape enlarges as we progress from the primordial to the primary to the secondary and to tertiary or Graafian. Only gets this particular thing further moves to this particular region further, like if we get this particular things develops into the fertilization. And we have to talk about this particular zona pellucida, little bit about what do we mean by the zona pellucida and Graafian follicle, which is I am saying like this is a kind of like a several times we see these questions are coming. Nowadays, it's very odd question to be honest, because they focus mostly on clinical questions, but still you can expect a Graafian follicle question as a easy questions which is coming as a Graafian follicle, or sometimes we get this particular thing after ovulations like corpus luteum or luteal glands, which comes as a physiology short note and as well as anatomical short note as well. What are the hormones that is liberated, etc., etc. What are the functions of the hormones, etc., etc., that we have to prepare accordingly. So, this is what we are going to see or talk about. This is how this particular thing in full zoom or high magnifications the mature Graafian follicle should be looking like. Oocyte with the zona pellucida with this radiating cells which is present in corona radiata and the antral follicles, and this particular theca cells, theca interna and externa. Externa becomes flat and which is highly high in hormone liberations they are producing, and a granulosa cells which is also modified form of follicular cells which are present to it. And this is what we call them as empty sac. After ovulation, this particular things is gradually regressing and shrinking, and white tissue is coming in here and replacing it into like a corpus lutea of albicans. So, this is what we are going to see in an ovary cross section picture. Germinal epithelium, tunica albuginea, cortex and the medulla contains this different stages of maturing ovarian follicle, primordial to this particular corpus albicans depending on the stages what they are coming on. And this Graafian follicle after ovulation becomes this hormone body, we call them as corpus luteum. So, histologically we are going to see this particular structure, and we have two of them. Split amongst yourself into two groups and see these particular microscopes and try to identify this outer cortex and inner medulla. And if you see any kind of like follicles, one of them have got like a mature Graafian follicles in it, try to identify them and take a picture and draw as well.